Let's see what you guys came up with. So I asked you, names parts of the body that have proteins. Muscle, skin, brain. DNA does not have proteins. Actually, I was surprised. I didn't think there would be one thing up here that didn't, but someone found one that didn't. DNA is nucleic acid, right? Not protein. Muscle, fingernails, heart, plasma. Well, plasma, not really. No. Stuff in plasma, yes. Nails, for sure. Muscle tissue, hair, blood, full, basically, body, basically, your whole body. Well, not your whole body, a lot of it, though. Head, shoulders, knees. Head and shoulders, knees and toes, knees and toes. Tête, épaules, genoux et pieds, genoux et pieds. That's the French one. Remember that? Did you guys do that in French class? You didn't do anything in French class? You're on the record for saying that. I just recorded it. I'm going to play that for Mr. Laurent. Muscles, everything, muscles. Pancreas, kidneys. Trick question, really, because proteins are pretty much everywhere, right? They're what make us up. They're our reason for living, literally. Folks, get on the same task. Every part of you works because of proteins. We have 30,000 different proteins in our body. Our DNA contains all the directions on how to make them. We eat carbohydrates to break them down into glucose, to turn that into ATP, a tiny nucleotide, to help us make more proteins and stabilize and keep the ones we have working. We break proteins down using the energy and the fat we eat in order to get amino acids to make more proteins. So really, you exist to make more and more proteins and sustain the ones you have. So today we're going to look at gene expression. That involves two processes. And if you're talking, please do not. I will call on you if you raise your hand. I swear to God I will. What role does RNA play in gene expression? We're going to talk about the three types of RNA. I'll remind you about them because you should already know about them. We're going to talk about what happens during transcription when you transcribe something. And we'll look at codons and how they determine the sequence of amino acids that result after translation. We might even play a game tomorrow. You never know. Okay. And then we'll look at, it is almost Halloween, right? So it's time to give you Halloween candy and junk food? Yeah? Okay, it's getting there. So then we'll look at the major steps of translation. Hopefully we'll get through most of this today. Probably not all of it, though. And then we'll look at what results from the expression of one single gene. We know that's a polypeptide, also known as a protein. So DNA provides the original information from which proteins are made in a cell, but DNA does not directly make proteins. RNA, or ribonucleic acid, is that second type of nucleic acid, which we've already talked about. It has the sugar ribose. It has the nitrogen-based thymine uracil instead of thymine, and it's single-stranded. It's much smaller. Okay, it doesn't have that helical structure. So it's way different than DNA, but it's still made of nucleotides, right? So gene expression is the manifestation of genes into very specific traits. When I say a trait and I say a gene, I mean a protein. Okay, my eyes are blue because of a brown protein, or brown. Oh, what did I don't even know what I just said. My eyes are brown because of a brown pigment protein. That's a lot of P's and B's in the same sentence. Okay. So gene expression produces proteins by transcription and translation. Two very specific words sounding kind of similar. They both start with a T. They're both kind of long, and they both end with T-I-O-N. Do not mix them up. They're very different. This process takes place in two stages, both of which involves RNA. I'm going to ask you, where does transcription take place at? I'll go back. Don't worry. I got a time machine. Where does transcription take place at? Where does transcription take place at? See if you remember. I'm jogging your biology memory. I bought 48 pumpkins yesterday. 48 pumpkins. I got 30-some kids coming to my house Saturday. The fall Halloween party. Oh. I'm going to get infested with small children. I hope it doesn't rain because it is not that big. It's not small, but it's not big. You know, he gives me a good deal. I go there every year. So, like $1. I think he sold me that many pumpkins for $60, so that's a pretty good deal. Pretty cheap, really. And they're pretty big ones. All right, let's see where we're at. I got 10 of you. I need more. Let's shoot for a dozen, at least. You guys, in about a week and a half, 2 weeks, we're all going to have a device in front of you, and I literally will not move on unless you participate. So start getting used to doing it. Even if you don't know, put IDK. Put an answer in. I'm getting sick of non-participators. What? That's like text language for I don't know, I think, isn't it? Yeah. IDK, but IDK, I don't know. It might be. IDK. Oh. Come on, Morgan. I almost had her laughing. It's too early for Morgan to laugh. There it is. Sam, I'm not even going to try. There we go. Okay. There we worked. All right. Let's see what you came up with. Wouldn't it be mean if I just moved on, didn't even look at them? The nucleus. Yes, for sure. Not the cytoplasm. Not the, don't pray. It's okay to pray, but you don't have to say God like that. That's like, you could use gosh or something. You're going to offend people in the world. You won't offend me, but you're going to offend somebody. It takes a lot to offend me. All right. So the nucleus for sure. Let's just talk generally about that. Transcription, in my own words, is copying DNA onto RNA. And we know DNA is stuck inside of the nucleus, so if you're going to copy DNA, you must be in the nucleus, right? It's like, aha, yes. Anytime you hear DNA, you must be in the nucleus. There's no other place it's going to be, all right? So, you're used to what? I know. Let's see if we remember the whole process. Let's go through it. So DNA is stuck inside of the nucleus. We copy DNA onto RNA. Wiggles through it. Goes out into the to find a to make a by assembling amino acids. The bond that holds amino acids together is called a peptide bond. We'll add little things here and there. Okay, so transcription is similar to copying or transcribing DNA, but think of it right now. You guys are copying notes off the board, and so the first stage of gene expression is called transcription, and that's basically just taking your DNA and making RNA. Now, all RNA is made this way, you guys. It doesn't matter if it's mRNA, tRNA, rRNA. It's all made this way. But when I talk about copying DNA to RNA, which type of RNA am I usually talking about? You guys got to stop blurting things out and jibber jabbering. We're going to be participating through that thing in front of you. Use it. What is the type of RNA that I'm usually referring to when I say copying DNA onto RNA? What type of RNA am I usually referencing? What type of RNA am I usually copying onto from DNA when I say we copy DNA onto RNA? What type of RNA am I usually talking about? There's 3 types. What is the one that I'm usually talking about? I know, it's warm in here, right? Yeah. They better get that fixed before the cats show up because you don't want to smell a warm cat. Yeah, 4th marking period. Yeah, well, mRNA, that's like backwards. Yes, mRNA, not tRNA, mRNA for sure, messenger RNA. Yep, mRNA. So that saying that I always say is the code is on mRNA. Everybody say this with me, and do not jibber jabber to your neighbor. The code is on mRNA. So there's a code in DNA, we know that. We copy the code onto mRNA. Code is written in code, right? So we don't understand it yet. We have to go through a translation process. That's out in the ribosome. But when you first copy it down, you're just copying the code. So imagine you're like sneaking into a building, and you got this secret message written in code, and you copy it down real quick, and you get out. You didn't have time to translate it. So you copy DNA onto RNA. What type of RNA? mRNA. Now, mRNA is what leaves the nucleus, right? Okay. You know what DNA says to mRNA every time it leaves? Don't forget to put your coat on. It's got the coat on. That was a smart joke, right? No? My kids are going to be so embarrassed by me someday. You just wait. You know? So transcription is similar to copying or transcribing notes from the board, just like you guys are doing right now. You might not understand it now. You're going to go home and translate it and study it, and then you'll get it, right? Yeah. Maybe. Yes, you may. There's a filtered water functional device over there. Oh, no. You got Ebola? What's going on? SARS? All right. Uh-oh. Here we go. So this, anybody scared of Ebola? I'm getting there. Your cousin is? So he came through and somehow made contact with someone? Oh, I'd want to be quarantined too. I would, because imagine if I came home and gave that to my kids, how terrible I'd feel. You know what I mean? I'd want to be. Yeah, that stinks though. That's scary kind of. I'm digging a big hole and climbing in it if it comes, and I'm taking my family with me till everybody's dead. What? No, it can't. But people aren't the biggest thing. Like if you bring a pen into a room to get something signed, you should toss that pen or burn it, right? And that's the kind of stuff that wasn't happening. And I think that's probably how it. Like when you sniffle your nose and you grab a pen, and then someone grabs a pen and then sniffles their nose. That's how these people are transmitting it. It is highly contagious through mucous membranes and blood and fluids. It's not a mildly contagious thing. That's what people don't realize. Like the fluid, yeah, you got to transfer fluid, but gosh, that stuff's everywhere. You're touching your table right now, and if someone touches that table where you were and then they lick their hand, that is. But it happens all the time. Have you met small children? They go like this. Then they grab like a candy, and then they go, here, try this, and then their buddy grabs it and they shove that in their mouth, and then girls are like, here, try my lipstick, and then they're like, oh, sure, and then they don't realize there's mucous membranes transferring all kinds of fun stuff. Yeah, but until I live with people who are smart enough to not do all of those things, I want them to be seriously quarantined. Like, I want them to stop letting people in. Why don't you just close down the border until you've taken care of your Ebola issue, right? So the second stage, we got off on a Ebola tangent there. So a little bit, but I got winter coming, and that kills a lot of stuff. So I'm counting on that. What does PSE stand for? So the second stage of gene expression. Hold on, guys. So we've talked about translation, copying DNA on RNA. Now we've got this coded message that we don't understand. We need to translate it. The second stage of gene expression is called translation. And this uses information in RNA to make a very specific protein. What is a protein? The assemblage of amino acids. What holds amino acids together? Peptide bonds. Okay. So translation is similar to translating sentences in one language, RNA, the nucleic acid language, to another language, the protein, the amino acid language. And I had you guys doing that lots and lots of times. Let's say it with DNA, right?
Oh, yeah.
Yeah, and you loved it. But here's the cool thing. You'll never wonder about that again. You know it. You're a pro at it, right? Yes, ma'am. You know it what? Best friends in Egypt? Yes. I am well aware of what I did not grow up in a cave, but I was born at night. So. Oh, I know. Oh, yes, I know. I know. I had a buddy that used to say what it really meant all the time, and so I understand thoroughly what it is. Okay, so we know DNA is this very large, you guys, I'm fighting your conversations, and we need to have one together, okay? It's on that thing in front of you, and it's with me. All right, so we have that very large structure, DNA, and DNA is stuck inside of the, it's not going to work. Here, we'll do this. Nucleus, right? DNA is too big to fit through that nuclear pore, so it has to stay in the cytoplasm. It would never fit through there. It's way too big. Let's just talk about how big, okay? DNA is 2.2 billion base pairs. Folks, we're going to reference DNA in size in here a lot, and we measure it in base pairs. How many G's with C's, how many T's with A's. So you have to understand, when I say 2.2 billion base pairs, how many nucleotides am I actually talking about? 4.4, right? So you're talking about 4.4 billion G's, C's, T's, or A's, and that's what your DNA is made of. Now, of that 2.2 billion base pairs, there are, and you should know these, 30,000 plus proteins, or otherwise known as genes, right? That's a lot of genes. It's like Levi on steroids. So you've got 30,000 proteins or genes on your DNA written in 2.2 billion base pairs. How many of these do you copy at one single time? How many of these can we copy at a time? Take a guess. I'm going to pear deck you real quick. That sounded kind of violent. I'm going to pear deck you. How many of these can you copy at once? How many of those genes do you copy DNA onto RNA at once? I didn't ask how many base pairs. I asked how many genes do you copy at a time? See what you guys are coming up with? Oh, that's creepy. What's going on? Anybody else feel kind of uncomfortable? Mr. Shaw. Two. Oh, you guys. Yeah, you're getting there. So one, right? We only get to copy one of those at a time. Now, the reason I talk about that is plain and simple. Let's really think about this for a second, folks. We reference that RNA is smaller than DNA, but until you actually think about how much smaller, you don't really understand the huge size difference. Because we show you these two drawings, and you look at this, you look at DNA, and you go, oh yeah, that's bigger than that, but not much. But really, a whole heck of a lot, right? We're talking about 1/60th the size, 1/60,000th the size, I'm sorry. Because again, you're talking about something that is 4.4 billion nucleotides, and so you're talking about something that is extremely small compared to DNA. One tiny bit of it. And there's one more piece of information that makes it even smaller that I'm going to tell you next chapter about how you splice RNA before you send it out to get rid of all the extra spaces. That comes in chapter 14, but it is even smaller than that. So DNA is super small compared RNA is super small compared to DNA. So we copy DNA onto RNA, mRNA, right? mRNA is written in these three-letter words. What are those three-letter words called? What are those three-letter words called? What are the three-letter words called that are on mRNA? What are they called? Three-letter words. It's okay to be wrong. Remember, put IDK, not BFE. Because we all know what that means, and that's bad. See what you have here. You guys are thoroughly entertaining with some of these things. Codons, yes, for sure. They are codons. Ions, no. Amino acids, not yet. We'll get there. IDK is a fair answer. Amino acids, no. Codons, yes, say it with. All right, so they are indeed codons. So there are different types of codons. I had you guys copied DNA codons onto mRNA codons and then onto tRNA anticodons. Okay, but you always read, you guys, mRNA. If you read DNA, you're going to be wrong. If you read tRNA, you're going to be wrong. You must always read mRNA to decode the message. Remember that chart from last year where you used the first, second, and third? No, I'll refresh your memory in a minute. So if you were to use tRNA anticodons or DNA codons, you would get the wrong amino acid. You need to use the mRNA codons. Oh, I apologize. Yes, I forgot. Some of you had me 2 years ago. That's like way too long ago, isn't it? Yeah. All right, so once RNA gets out of the nucleus, it goes and finds a, everybody say ribosome, out in the cytoplasm, right? And then that ribosome can make a protein by assembling amino acids, by forming peptide bonds. Yay, okay. So all of these steps in gene expression involve RNA. In cells, there's 3 basic types of RNA that complement DNA and translate the genetic code into proteins. What are proteins? They are large polypeptides that function, basically. What's the word poly mean? Many, peptide referring to the bond between amino acids. So like DNA, RNA is made of nucleotide subunits linked together. And RNA differs from DNA in 3 basic ways. First, RNA usually is composed of 1 strand of nucleotides rather than 2 strands. Why do I say usually? Not always. Sometimes RNA folds on itself and then bonds to itself, so in certain places it can be double-stranded, like in tRNA. We'll talk about that later. Oh, I'm sorry. I'm sorry. Hay is for horses. I do. No, I've got one. I do have a pony. And then we have mango now. And we got rid of the big ones. No, the pony's a Shetland, but then the mango is a big quarter horse. He's actually 17 and a half hands. I used to have giant draft horses. Because my daughters wanted riding horses. I got pictures of them up there, actually, so you can see them. Not right now, though. Hans and Cecil. They're in Wisconsin now on a kids' ranch doing hay rides. So second, the RNA nucleotide contains the 5-carbon sugar ribose. Anytime you see a word that ends with O-S-E, it is a sugar, right? Okay, sugar. Rather than the sugar deoxyribose. So the thing you need to realize is both of these are 5-carbon pentose sugars, but if you have ribose, you're talking about ribonucleic acid or RNA, and if you have deoxyribose, you're talking about deoxyribonucleic acid or DNA. And thirdly, the RNA, can you say thirdly? I just did. Third, the RNA nucleotides have a nitrogenous base called uracil instead of the base thymine, which kind of can mess you up sometimes if you aren't paying attention. So you normally, if I say A, you go A goes with T, but if you're going from DNA to RNA, then A goes with U, okay? So keep in mind, if I give you a test question, a lot of times if I ask you or tell you you're going through the transcription process and I give you T's as possible answers, you know those are wrong. You're looking for U's, right? So uracil is complementary to adenine whenever RNA pairs with another nucleic acid. So when we start to copy, not like yesterday with A going with T, A has to go with U when you're making RNA. Even when you're going from RNA to other RNA, it's still U that replaces the T. Bless you. So the three main types of RNA, oh, let's do this. Let's do this. Let's make sure we get that. This will be fun. I always forget we have these. So I'm going to have you guys draw me, or make, I should say, a complementary strand for this DNA strand. Give me the mRNA that would transcribe. Oh, I got a really bad joke I just made up in my head. I can't even tell it. It's so bad. What did RNA say to DNA when he caught him cheating or copying? AU. See, it's terrible. Because A with you. Oh, I'm sorry. Here, I got to show. Let's see what you came. Let's see what you're coming up with. Wet. Wet. What is that? Well, we got to work on. We got to work on a few things there. Oh, somebody's in. Is that Mr. Pellis? Is he in here? Okay, I got to actually go back and see what I wrote because I don't remember. So it should be U, A, U, C, G, G. That's the right answer. Oh, geez. There we go. All right, so hopefully you put, This is DNA. This is mRNA. Yep. Keep going.
On that sheet, it said the mRNA was the first one we copied from the DNA sequence, and that was T.
No.
Yeah, it was.
No. It said the DNA codons, not the RNA codons. Look closely. There's such thing as three, and that's the thing in this class is a little different. There are DNA codons, there are mRNA codons, and then there's tRNA anticodons.
Because I looked at that sheet, and
I know, because it says
the mRNA does not have
mRNA always has U's, not T's. All RNA. I'll take a look at it with you, but I'm thinking you might have misread something. Okay, so then when we look at T, let's go one step further. If I took this to tRNA, it would be A, U, A, G, C, C. So I would not want, though, you guys, to use tRNA anticodons to code for my amino acid. This is what I would read on the chart. These three-letter words. Those three-letter words are called codons. Oh, that was kind of a C. Codons. Okay? Yeah, I don't know. That's too much effort. Oh, okay. Come on, Bluetooth. There we go. All right. Oh, we did this one already, didn't we? Yep. So the three main types of RNA which play a role in gene expression are mRNA, messenger RNA, and yes, you need to know that they're abbreviated like mRNA, right? tRNA, transfer RNA, and ribosomal RNA known as rRNA. So messenger RNA, or mRNA, is produced when DNA is transcribed into RNA. And mRNA carries instructions for making a protein from a gene and delivers the instructions to the site of translation. The code, oh, that's not an O. Code is on mRNA. Oh, work, you silly machine. There we go. So that's why I always say mRNA has the codons, right? The code is on mRNA, codons. Is this little slogan for today? This is brand new in Pear Deck. They started putting the code on. I like that it's on there. I don't have to write it on the board over here anymore. But it is, isn't it? That's brand new today. Because I've been having to write it on the board every time, and it changes every hour, and it's frustrating me, right? I don't have to put it in BFE anymore. So at the site of translation, tRNA or transfer RNA reads the instructions carried by mRNA and then translates the mRNA sequence into protein subunits called amino acids. So tRNA has what is called the anticodons. I think we know that. And then there's this other one. It's called ribosomal RNA. Bless you, I don't make you remember anything about rRNA except that it is what ribosomes are made of. So if I ask you, what are ribosomes made of, you should tell me proteins and rRNA. Are you sure? Yes. It's the pirate RNA. What? R. In 5 years, how bad will the jokes be? That's the question. They're going to get really bad. This is after 12 years. Imagine 5 more. Yeah. I'm a dad. I got 2 kids. So ribosomes are the cellular structure where protein production occurs. We understand that. We copy DNA onto RNA. RNA wiggles through a nuclear pore. What kind of RNA is that? mRNA. mRNA has what on it? Codons, which are the 3-letter words which code for amino acids, right? So mRNA wiggles through a nuclear pore, goes out into to find a ribosome to make a protein by assembling amino acids. A lot of amino acids together are called a polypeptide, held together by peptide bonds. Okay. I'll have you all brainwashed by the end of the year. That's my goal. So you go to college with this one. Folks, if you take that out of your biology class in high school, I'm going to tell you you're ahead of most people, okay? Which is scary, because it's not that hard, really. So during transcription, the information in a specific region of DNA or a gene is transcribed or copied onto mRNA. Transcription is carried out by a protein called RNA polymerase. And you go, oh no, Mr. Mason, that sounds just like DNA polymerase. And I say, yes, it does. The fool that named that was a fool. He should have called it something very different. The reason why is this is the biggest misconception. Kids mix up transcription with DNA replication all the time because this enzyme sounds extremely similar to DNA polymerase. Okay, it is a polymerase enzyme, but this time we're making a strand of RNA instead of a strand of DNA. Okay? So you have to pay very close attention when I write either RNA polymerase or DNA polymerase. If you say RNA polymerase, I'm talking about transcription and making codons. If you see DNA polymerase, I'm talking about making another strand of DNA. So transcription begins when RNA polymerase binds to the specific DNA sequence in a gene that is called the promoter. What do promoters do? They get things started, right? Okay, so I'm looking at DNA. I'm an RNA polymerase, and I'm like, oh, there's a start sign, says promote here. So I start copying right there, okay? Now, RNA polymerase then unwinds and separates the 2 strands of double helix to expose the DNA bases on each strand. Now, this is different. Who, back in DNA replication, split DNA in 2? Who was it? What was it? I should say, not who. What was it that split DNA in 2 during DNA replication that is now done just by one enzyme? Yes, please. Por favor. I've got that French head and shoulders, knees and toes playing in my head nonstop from high school. That's how bad my morning is going right now. Over and over again. You ever get one stuck in your head, Miss Raymond? Not in French? What was the other one they used to make us sing? What was that other one? Can't remember. What'd you guys put? Oh my goodness. Beyonce around. All right. Let's see what happens. Oh, my Lord. Beyonce's breaking hydrogen bonds. All right, not really. Okay, DNA helicase, yes. Hershey and Chase, no. Helicase, yes. Helicase, yes. Not DNA polymerase. Remember, DNA polymerase is not the one that splits it. So, you guys, the main thing I want you to pay attention to here, yes, helicase, okay, is that there is no helicase, folks. There is no helicase in the process of transcription, which is way different than DNA replication. Remember, DNA replication had two enzymes, DNA helicase on zip DNA, then DNA polymerase reformed new nucleotide hydrogen bonds and covalent bonds on the side chains. Now we're talking about just one single enzyme, and it's RNA polymerase. It does everything. So RNA polymerase then unwinds and separates the two strands. And then RNA polymerase moves along the bases on the DNA strand and adds complementary RNA bases as it reads the DNA of the gene, copying those A's to U's and T's to A's, making those codons. So then as RNA polymerase moves down the DNA strand, we start to see a single strand of mRNA growing out of it. Now behind the moving RNA polymerase, the two strands of DNA close up and reform the double helix. Why do you think we need to close DNA back up? Why do you think we need to close DNA back up real quickly? Why do we need to close DNA back up? What if I gave you a backpack and ripped all the pages out of your book and threw them in there? That's a hint. And that was your book for the year. Why would I need to close DNA back up? It would all be there, but it wouldn't be what? It'd still all be there. Able to be. See what we came up with. Okay, not to prevent mutations. To keep everything in the same place. That's not far off. To keep things in order, right? So it doesn't unwind for sure, right? Because those hydrogen bonds would definitely break. Mutations more than likely would not happen, although you could get a translocation. We'll talk about that more later. So it doesn't unwind, kind of. I mean, well, escape, no. To avoid damage, you're not going to damage it. Run away like my dog. No. It's too big and everyone get lost. And do not hurt my books. I don't know either. I'm working on that one in my head. Prevent mutations so it don't leak. All right. IDK is an honest answer. All right. So how about this? We talked about how big DNA was, right? Folks, this is an important concept to really understand. If I were to ask you to read 5 pages of your book at the same time, could you do it? No. And so you only open your book to the pages you're going to read. And if your book was just open like a accordion all the way around, RNA polymerase cannot do all those pages at once. Here's the question though. Could I have more than one RNA polymerase on the same DNA strand transcribing at the same time? So I could have a lot of different readers on the same scroll basically, but they'd have to close their section up so when someone else comes along it's all organized, right, and in order. Remember, we're talking about 2.2 billion base pairs. We're talking about 30,000 genes worth of information, and we only read one at a time. So just keep it into perspective as to why you would not want your book just all the pages spread out in front of you and open all at once. You can only read so much at a time, and you have to keep it organized because are you going to make more than one hemoglobin protein in a day? Yeah. You guys want to know how many you make in a minute? 1 trillion. 1 trillion hemoglobins in a minute. It carries oxygen in your red blood cells. It's a protein. Everybody stand up real quick. I got a couple of you that look like zombies. Stand up. Stand up. She was about to drool on herself. Her eyes were almost shut. Stand up. I'll do it too. Since we're talking about head and shoulders, we'll do it. Here we go. Yep. All right, you guys ready? Everybody touch your head. Say tet. Tet. Touch your shoulders. Say posure. Say posure. Touch posure. Posure. Touch your knees. Say genoux. Genoux. Touch your feet and say pied. Touch your knees and say posure. Genoux. Say genoux. Genoux. Touch your feet and say pied. All right, reach up, touch the sky, make up a word because I don't know what that one is. Spirit fingers in French. All right, sit down. Oh, that felt kind of good, actually. We should do that every day. Oh, just me. Mr. Mason, look like an idiot just by yourself. I don't. I don't know the rest of it, though. In Arabic, yeah, right. I don't know. I don't know how that one works. All right, so, where did we leave off? Oh, we're done there. Okay. No, we're not. I didn't mean that. I said that out loud. All right. So, transcription is not the same process as replication. I thought I did. No, we did get that. We got that. Oh, I will leave it up for you. Okay. Oh, who said that out loud? You're supposed to say that in history class or something. I did. I just got up and did head and shoulders, knees and toes in French. Oh, okay. Well, here. You want more pictures? I got some coming. Don't worry. They're coming. They're coming. I've got the video we still have to finish from yesterday. So transcription is not the same process as replication. Although, folks, it is easily confused because they're both dealing with copying nucleic acids. They're both dealing with enzymes, and one of the enzymes sounds very similar. There's polymerase in both of them. In transcription, a new molecule of RNA is made from DNA, and in DNA replication, remember, we're making a new molecule of DNA from DNA. So, there's your picture. You ask and you shall receive. Lexi Rao. All right. So, let's talk about what's going on in transcription. Why do we call it transcription? Because we're copying DNA onto RNA. Okay? So let's work through this together. Here's DNA. This is the promoter region that RNA polymerase will read. Once RNA polymerase reads a promoter, it will start to copy and unwind DNA. The RNA polymerase will then unzip DNA only at the section it is at. After it is done, it's going to reform DNA back into a helical structure. It's almost like a nut that's going down a screw because it's basically a nut on a spiral staircase in a way, okay? Because it's going around and around and around. Remember, DNA is a helix. It'd be like you on one of those little slides going down DNA, okay? So RNA polymerase is going to break hydrogen bonds. When it does that, you guys, it is going to expose DNA. Now, you really need to pay attention here. We can only read DNA one way. We talked about that yesterday. When I make mRNA, do I copy both sides or just one side of DNA? One. Let me explain that. I'm only unzipping DNA in a small section. So I'm only copying the template side of DNA, the side that reads from left to right, from leading to lagging. We'll talk about that later, 5' to 3'. As I read DNA and add those nucleotides on, I'm going to get this single strand of RNA. And this is why RNA is single-stranded. We only copy one side of DNA at a time. Once it reads the termination signal, it stops copying, and at that point, that single strand of RNA is released. Now, there's more to this whole process of making RNA. We're going to talk about it in Chapter 14. But for right now, you need to understand that when you make RNA, you're just copying the template strand of DNA onto RNA. How is this different? How is this different than DNA replication? Please tell me. How is this different than DNA replication? This is where most of my test questions will get missed, right here. How is this process different than DNA replication? Please type something into that device glowing at you in front of you. Respondez-vous s'il vous plaît. There's more French. Five responses. Nine responses. Come on, let's get a dozen. If it takes a minute, it's going to be too long. Come on. See what we came up with. And I'm waiting for 12. Bon anniversaire. That's happy birthday in French. All right, here we go. Sweet, chocolate. I don't get that. Okay. DNA replication does not need a promoter. That's very true. Okay. DNA replication involves two enzymes. That's a good observation. I'm going to lock this out, not to stop anybody from writing, but just so it holds still. Let's see. Different enzymes in DNA replication, helicase and DNA polymerase. Very true. And a new strand of DNA is created while transcription results in an entirely different molecule, for sure. DNA replication involves two enzymes. See if there's anything new. No call ahead, no waiting. Okay. Well, I don't know what that says. DNA replication does not need a promoter. Yes. DNA replication is different because there's no RNA involved. Very good. So in replication, the one enzyme has to loop the DNA around on one side to be able to read after the write, but not for transcription. That's true because we're only copying one side, and in DNA replication, we're copying both sides. DNA replication makes two new strands of DNA. Yep. Complementary RNA is added for sure. DNA transcription makes RNA. Yep. Doesn't create a new strand of DNA. I see you too. It's only copying one strand. There isn't a new strand of DNA. You are not replicating DNA. You are copying DNA for RNA. I don't know how to make sense of that one. I'm working on it. It's like a thought, but it's a good thought. There's no such thing as a bad thought, right? Well, I guess we could argue that. So at any rate, you guys understand the difference between DNA replication and transcription. This is a point of confusion. I just want to make that loud and clear. So this leads into those three-letter words that are written on mRNA. A three-letter nucleotide sequence, remember, is what is called a codon, and each codon corresponds to one of the 20 different amino acids. And codons act as a start or a stop signal for translation. There are 64 mRNA codons, and each codon specifies only one amino acid, but several amino acids, bless you, have more than one codon. The system of matching codons and amino acids is what is called the genetic code, and the genetic code is based on codons that each represent a specific amino acid. So when we talk about encoding DNA, we're talking about the process of translating mRNA into proteins. Now what I want to do is I want to go back to that video we showed yesterday, and I want to take you from DNA replication through transcription so we're ready to go through translation tomorrow, okay? So we're kind of done taking notes, but I don't want you to stop thinking because I really want you to watch this with me and pay attention.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones.
Please do not get anything out.
The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again.
So, I know we've been talking about polymerase, but think about what polymerase has to actually do. It has to wind around this whole structure of DNA. Oop, my drawer's not going to work. There. This whole structure of DNA, and it has to, almost like it's going down this long, slippery slide, it has to go all the way through DNA and copy it. And it has to rewind this supercoil and turn it back into a condensed structure. Otherwise, your DNA gets all disorganized.
Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
What process is that called? Mitosis. What's about to happen right here? Cytokinesis. Yay, okay. Let's check. This was yesterday, DNA replication. 2
Using
enzymes
computer
involved.
animation based on molecular research, we are now able to see how DNA is actually copied in living cells.
Helicase in the middle.
You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
So this is what we did today now, transcription. That was 2 enzymes.
What you are about to see is DNA's most extraordinary secret, how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene.
What is that bundle of factors called? A promoter. Okay, promoter.
A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands.
So could I ever copy both ways with only unwinding in that little section? No, so just keep that in mind. That's why we copy one side.
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin.
So what do we call this? mRNA. What are the 3-letter words written on mRNA? Codons. What do they code for? Amino acids.
DNA. called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene.
Okay, tomorrow
The only difference is that in the RNA copy, the letter T.
Don't forget to study for your vocab test that will be on Thursday. Well, yeah.